Okay, let's go ahead and get started here. Page 277. And yes, I understand we have individuals who are gone, but we still keep going. Page 277. Okay, so on page 277. Okay, page 277. The inheritance of traits. Okay, Mendel noticed certain varieties of garden pea plants produced specific forms of a trait, generation after generation. So these would be some of the examples that he had observed. We just talked about this one yesterday. So we thought we'd give you some more varieties to pick from. For instance, he noticed that some varieties always produce green seeds and others always produced yellow seeds. In order to understand how these traits are inherited, Mendel performed cross-pollination by transferring male gametes from one flower to a true-breeding green-seeded plant to the female organ of a flower from a true-breeding yellow-seeded plant to prevent self-fertilization. Mendel removed the male organs from one flower of the yellow-seeded plant. Mendel called the green-seeded plant and the yellow-seeded plant parental generation, also known as the pea generation. So on the next page, you can see, we said this yesterday, we often just look at any type of garden variety of different types of peas as vegetables. We always think that they're dominant being green. But green is actually the recessive trait, as you can see on page 278. Okay. We're not too concerned about the F1 and F2 generation, so genes in pairs. Mendel concluded that there must be two forms of the seed trait in pea plants, yellow seed and green seed, and the alleles defined as alternative form of a single gene passed from one generation to a generation. Can I please continue? All right. Please. Okay. Therefore, the gene for yellow seeds and the gene for green seeds are each different forms of a single gene. Mendel concluded that the 3-to-1 ratio observed during this experiments, he called the form of the trait appeared in the F1 generation dominant, and the form of the trait that was masked in the F1 generation recessive. In the cross between yellow-seeded plants and green-seeded plants, the yellow seed was the dominant form of the trait, and the green seed was the recessive form of the trait. Okay, so anytime we're talking about dominance, it's what is expressed in the cross. So, like for humans, when it comes to eye color, which one is dominant? Brown. It's brown. Okay, blue is actually recessive. Okay. So anytime we're putting these terms together, if it's homozygous recessive, it's two little letters. If it's heterozygous, it's one of each. And if it's homozygous dominant, it's two capital letters. Okay, so last thing we want to go over before we apply some of this content here. Genotype and phenotype. Okay, so what we're looking at here, Mendel used homozygous yellow seed green plants in his cross. Okay, oops, I went too far. A yellow seed plant could be homozygous or heterozygous for the trait form. The outward appearance of an organism does not always indicate which pair of alleles is. In the case of plants with yellow seeds, their genotypes could be YY, two capitals, or capital Y and lowercase y. The organism's allele pairs are called the genotype. In case plants with yellow seeds, their genotypes could be capital Y, or capital Y, lowercase y. The observable characteristic or outward expression of an allele pair is called a phenotype. The phenotype of a pea plant is with the genotype YY will be green seed. So we observed that yesterday. We just did not really realize that's what was happening. So we're going to go through these different types of crosses, whether they're monohybrid or dihybrid. Okay, so on page 280, they talk about monohybrid and dihybrid. So a monohybrid cross, other than tall and short, I want you to pick one of those traits. No, I said other than that. Okay, so since we just read that, what does it mean if it is heterozygous yellow? No. No. We just read it. We just told you. Okay, if you said you want to do yellow and green, what does it mean if it is heterozygous yellow? I can't hear you. True. Yeah, it'd be a capital Y and a lowercase y. That is heterozygous. So, and it's just personal preference that we put a line over the top. You don't have to do this. It just helps me indicate which one's the recessive trait. So, we have this yellow colored pea plant that is heterozygous. What do you want to cross that with? Another heterozygous? You want to cross it with a homozygous yellow or homozygous green? Okay? So if we say we cross this with a green plant, we know that this is dominant. Okay? We just read that. And then this is green. What is the only two alleles that can make this green? Then it's yellow. Two lowercase y's. Okay? So now, just like what we did yesterday, go ahead and do that cross, and then you give me the genotype and phenotype that we just read. Okay? Okay, go ahead and do that. We'll work on it together, but I want to see how well you are doing, because we just read all the content that you need for this. Okay, so we had this filled out by one of your classmates. You can tell that's not my handwriting there. And what we see happening here is now we want to look at the observable traits and what the genetic makeup is. So what that means then, what is our genetic makeup? In other words, okay, what type of alleles? An allele is just a single letter. So you need to pick out a combination. What is a combination of letters that we see here? Okay, you need to be a little more specific because they're all Y's though. Capital and lowercase. Okay? Capital and lowercase. Okay? And how many of those do we see? You actually see two of those. Okay? So we're observing two of those. Here's one, here's one. Okay? So then we need to have four combinations, we said. We've got two of them. So what is another combination that we see? Okay, so lowercase y and lowercase y. And how many of those do we see? There's two of those as well. So what that means then is these numbers have to add together to what is inside your box. There's four boxes. 2 and 2 gives you your 4. All right? No, because you want... Okay, now you can go pick that up, please. No, it hit the tape. Well, maybe it bounced off into there, but...
Yeah, it went in the trash can.
Okay. That's fine then. So, I'm sorry, what was your question now?
Like, so you would have wrote it out as like 2 yellow and 2 green. That'd be right.
For the phenotype, yes, but we want the genetic makeup. Okay? The genotype is the genetic makeup that we see, the different combination of alleles. The phenotype, on the other hand, that's why we put a P here. Okay? So, what would you observe with these two alleles? Now you can do your color. I know that's capital, that's lowercase, but what does it look like?
Looks like a yellow.
We have 2 yellow.
Yeah.
Okay? Because that dominant trait is present, it will mask over the color. Okay? So then we've got 2 yellow. And then what is this genetic makeup then? It'll be green. So you have 2 yellow, 2 green. Okay? No. I didn't quite I heard your question. Does this always turn out the same? Was that your question?
Or like, for our genotype it says like 2 and 2. Will that ever say like 3 and 1, and then our phenotype will say like 2 and 2? Or will
It can, but you don't know that until you do this. Okay. Do you think you need to see another example of this?
No.
So this is a monohybrid cross. Mono meaning just one trait that you're coloring, or one trait that you are testing. Seed color, that's all that you're doing. That's why it's a monohybrid cross. But when you look at the next stage on page 280, there's also dihybrid crosses. That's where you're testing 2 different traits. Can we erase this?
Yeah.
Okay? So we're starting this dihybrid cross on page 280. These are the traits that we picked. It says, what is the phenotypical ratio of a homozygous tall wrinkled pea plant with a homozygous recessive pea plant that has that for height, and it's homozygous round pea plant? So what that means then is tall is dominant over short. Okay? And round is dominant over wrinkled.
Can wrinkled ever be over round?
No. It's just the way it is. I mean, that's just the way the genetics works. I mean, the frequency that it happens.
How do you know if there's one capital letter or 2?
If there's one,
Capital letter or 2?
you're always going to use 2 letters. If it's
How do you know if one's capital and one's lowercase?
if it's heterozygous or homozygous. Okay? So what this, in case what you're wondering, this might be rounded over wrinkled. Okay, so we have everything we need. So when you turn the page to 282, you see this is not a 2x2 square. This is a 4x4. Okay, so you need to make this quite a bit bigger. So we will go ahead and set that up.
You spell wrinkled wrong. Thank you for fixing that.
You are welcome. I just didn't have the right color marker. I can do a better job than that. Okay. So there's our 4x4. And what we need to do now is set up our cross like we did similarly with our monohybrid cross. So let's look at what we are testing. Okay, so you always want to make a key for this. So capital T for height would be tall. Lowercase t would be short because that's what it says. Capital R would be round. And then lowercase r would be wrinkled. Okay, so here's your key. You're always going to want to make a key for this. Okay, that meaning it gives you your alleles and what it is that they stand for. Now, this happens when we're talking about a complete dominant cross. Tomorrow we might get into your codominant and maybe incomplete dominant. The reason that that's important is the best way to see that is through different coat colors of cattle. Okay, so you might, but you don't have any cattle, but you might know this.
Oh, we have cattle.
Oh, okay, you do. Okay. So, what coat color is a Hereford? And it's a red-white face. So then, if you have a black Angus, then, which is just all black, okay? So if you cross that black Angus with a Hereford or red-white face, both traits get expressed at the same time. You get a black-white face. So you see the black body of the Angus and the white face of the Hereford. So when you, going back to your question over here, do these ever get flip-flopped? And the answer is no, just like for the same reason. You always, always, always get a black-white face when you do that cross. It's just the way nature selected genetics to work. Okay. So then we see homozygous tall. What are our two alleles for homozygous tall? Capital T and capital T. And then what are our two letters for wrinkled?
Someone threw a paper at me. It was very,
Okay.
came from that way.
So what is wrinkled? And? Okay, two lowercase r's. Okay. There's one parent. Then with a homozygous recessive for height. So what two alleles are those? Two little t's. Okay? Two little t's. And then it says homozygous round plant. What are our two alleles for that? Two capital R's. Okay?
I already know that.
Okay? Looks to me like I'm thinking that, is it possible that we're going to have 16-0 here? Are we just agreeing? Okay. We're going to get different types of variance in the alleles, but I think you can make a prediction that it's going to be 16-0. Okay? So let's go ahead and take a look at this. This is where you want to do a similar process like this. Okay? And I always do this every year. This isn't just unique to this year. When you FOIL these out, what does FOIL mean to you when, let's see here, okay. You do that in algebra probably quite a bit, right? When you are factoring something or you FOIL it out. You do the first, the outside, the inside, and the last. That's what FOIL means, right? Is that correct? Okay. So that's where this comes from. So on the left side here, our first term, when we FOIL that out, capital T and what? Capital T and lowercase r. And maybe that's what you said. I didn't quite catch all of that. Okay? Then we want to do the outside. What do we have? Capital T and, okay? Now we do the inside. What does that give us? Capital T, lowercase r. Then we do the last, which is, coincidence enough, capital T, lowercase r. Okay? So if we FOILed out this side, we're going to do the same thing over here. What do we have? Lowercase t and capital R. Then we do the outside in our FOIL, and we've got lowercase t and capital R. Okay? Then we do the inside, right? That would be this one and this one. Lowercase t and capital R. And then we do our last one. Lowercase t and capital R. Okay? So if you're having, and maybe some of you are having problems with this, they go through this, the author does on page 282. You always have that option. You can go back and read this in the book, and it will give you the same content. Okay. All right. So as we look in here, we made that prediction. You can't do this very often with that of a dihybrid cross, but when this comes to masking over, what's going to happen with the height? Is that dominant trait going to be present in each and every box? Yes. Okay. Is that wrinkled trait going to be present in each, or excuse me, is that round trait going to be present in each and every box? Yes. Okay. So we already know that it's going to be 16 to 0. Okay. Like I said, that doesn't happen very often, and I would not recommend doing that, just saying, I'm just going to guess and say it's going to be 16 to 0, but sometimes it works out that way. Do we believe that, or do we need to go through it?
I think the most confusing part is the FOIL.
Okay, well, and that's why, and maybe this is meant to help you. Maybe it doesn't, okay? Okay, so then it's just like you're distributing that in algebra.
Multiplying
Okay,
the first and third, like I don't know, something about the first and the third, and I don't know, you're just multiplying them all by each other.
okay. So what do we have inside this box? Keep your letters together. Capital T and what? We said keep your letters together, though.
20 for 20 minutes, that's fair. No, I'm just kidding. I have nothing on that.
Would that be what it is? It would be. Then what about in this box? Capital T and lowercase t. And if you put these, you don't have to put these lines over it, but it's sometimes useful to keep the dominant and recessive trait separate. Okay? Then what about our R's in here? Capital R and lowercase r. Okay? So let's just pick a random box like this one. Okay? What would be inside this box that we have labeled as X? Capital T, lowercase t, capital R, lowercase r. Okay, maybe we can pick out another one at random. How about this one? Same thing. Do we see a pattern that is emerging here? They're all going to be the same. Capital T, lowercase t, capital R, lowercase r. That's one of the things that happens when it comes to genetics. Okay? If you just throw one letter off, it throws the whole thing off. Okay, I mean the phenotypical ratio. So I'll leave that for you to finish, but we already said that this is all going to be the same. So tomorrow we'll continue with some of these dihybrid crosses. All right, so we'll work on your vocabulary next week. We'll probably have an evaluation, the first one, because there's a lot of terms. Okay? All right, so that's all I have. We'll catch up to you next time.